Hi students, today we will learn about physiological anatomy and functions of cerebral cortex. The brain is one of the largest and most complex organs in the body, and it is composed of billions of neurons that will communicate together by forming numerous connections and synapses. There are three main divisions called cerebrum, cerebellum and brainstem. The cerebrum consists of two cerebral hemispheres. The outer layer is called cortex. It is grey matter and the inner layer is having white matter. The brain weighs 2% of the total body weight and it receives 15% of the cardiac output. See the embryology of the cerebral cortex or pallium. The first step is the patterning. Second step is the neurulation. Third step is the neurogenesis. Fourth step is neural migration. And the fifth step is the synaptogenesis. And sixth step is the synapse structure formation and its stabilization. First coming to patterning. The basic plan of the mammalian brain is laid out at the neural plate stage only. At this stage, signaling centers that surrounds the neural plate produces some signaling molecules. Some are FGFS, BMPS and SHH. The combinations of these signals will confer specific regional fate on the neural plate cells. Next coming to neurulation. The edges of the neural plate folds towards each other and fuse together and thus forms a neural tube. At the earliest stages, the neural tube contains three primary brain vesicles. The neuroepithelial progenitor cells divides and rapidly they will expand and form pool of the neural progenitors. Then the neuroepithelial progenitor cell subsequently transforms to other form of progenitor types, primarily radial glia. They will form glial cells. Then comes neurogenesis, in which cortical neurons are born. As development proceeds, an increasing proportion of the radial glia divide, generating large number of neurons. And another important population of neural progenitors known as intermediate progenitors or apical progenitors are found in the subventricular zone. The intermediate progenitors then continue to divide and makes more and more neurons. Then coming to neural migration, the neurons migrate radially towards the outer edge of the embryonic cortex, which is guarded by scaffold which is provided by radial glial fibers that project from ventricular edge to the pial surface. Neurons in each cortical layer have distinct molecular signatures associated with their specific functions. Next coming to synaptogenesis. This is the first step in neural circuit assembly. The cell adhesion molecules such as neuroligands and neurexins are recruited to the sign of the future synapse where they form a bridge between the axon and dendrite. This initiates protein specialization to organize the active zone of the presynaptic terminal and postsynaptic density. During this process, scaffolding proteins such as membrane-associated golanate kinases are recruited to the site of axodendritic membrane contact. Then comes synapse stabilization. Thousands of synapses and dendritic spines are formed as per one neuron. They are added, but the majority of the synapses are removed or withdrawn, and neuronal activity plays a key role in this. Many of the proteins located in the developing PSD, that is postsynaptic density, play a role in synapse stabilization, as many were shown to regulate the synapse number and size. Neuronal connections could be between neurons from other brain structures that are further away, that is long range connectivity, or with neurons from the same region of the brain, that is local connectivity. These connections are not final, as many connections are made throughout the embryonic and early development of the brain, which will be refined later on in development as connections that are used more are strengthened, that is activity based neural connections, while connections that are used less are pruned. You see this CNS development. This is prosencephalon, this is mesencephalon, and this is rhombencephalon. Prosencephalon is again divided into two parts, that is telencephalon and the diencephalon. The telencephalon forms the two cerebral hemispheres and it has a cavity, that is lateral ventricles. And this diencephalon forms thalami and etc. some parts of surrounding the thalamus, and it has third ventricle, the cavity. Then coming to mesencephalon, it forms the midbrain and it has aqueduct cavity. This rhombencephalon is again divided into two, that is metencephalon and myelencephalon. The metencephalon forms pons and cerebellum and this myelencephalon forms medulla. It has fourth ventricle as its cavity. Then this medulla is continued as spinal cord. Next coming to phylogenetical divisions of the cerebral cortex. So phylogenetically, first, first one is allocortex or limbic cortex. It constitutes about 10%. Allocortex or limbic cortex. In this you have archipallium and paleopallium. Archipallium is composed of hippocampus and a dentate gyrus. Paleopallium is composed of uncus and a part of parahippocampal gyrus. Okay, this is archicortex, this blue colored one. Okay, it is composed of hippocampus and dentate gyrus. This is paleocortex. It is composed of hippocampus. It is composed of uncus and a part of parahippocampal gyrus. And it has 3 or 4 layers of neuronal cell bodies in contrast to 6 layers of the neocortex. So in everywhere you have 6 layers in the neocortex, but in allocortex you have only 3 or 4 layers. Then coming to mesocortex. It is the transitional zone between the allocortex and neocortex. So this includes cingulate gyrus, a part of parahippocampal gyrus and subiculum. Okay, cingulate gyrus, a part of parahippocampal gyrus. This is cingulate gyrus. Okay, and a part of parahippocampal gyrus and subiculum. Then comes neocortex or isocortex, which is 90% is present in the brain. So this is neocortex. Then coming to train brain theory or a three-parted brain. So our brain is parted into 3 parts. That is one mind and it has 3 brains. Means neocortex, that is helpful for language, thought and verbal expression. And limbic system, it is for language, emotion and feelings. And basal ganglia, it is for language of the body, sensation and impulses. So this basal ganglia is well developed in the reptile brains. Limbic system is well developed in the mammalian brain. And this neocortex is well developed in the Homo sapiens brain. So they will perform intellectual and executive functioning, verbal language, conscious thought and self-awareness. You see here, this is neocortex. Okay, neocortex. This red one is the limbic system, and this is the reptilian brain, that is basal ganglia, brainstem. So you see the three-parted brain. The human brain is like this and it contains mostly neocortex. So this is helpful for language, abstract thinking, imagination, consciousness, reasoning and rationalization. Okay. This is mammalian brain. Mostly the limbic system will be well developed. So they can show emotions, memories, habits and attachments. Then coming to, for example, lizard brain. So here the brainstem and cerebellum are well developed. So they can do autopiloting, fight and flight. So this is D. McLean's model of the triune brain or three-parted brain. So this triune brain is a model of the evolution of the vertebrate forebrain and behavior, which is proposed by American physician and neuroscientist Paul D. McLean in the 1960s. Then coming to the external features. It has two cerebral hemispheres. Okay. They are separated by falx cerebri and corpus callosum, and it has three poles: frontal pole, occipital pole and temporal pole, and it has three surfaces: superolateral, medial and inferior, and it has three borders: superomedial, inferolateral and medial, and it has sulci and gyri. You can see here, this is corpus callosum. This is medial longitudinal fissure. One is the left hemisphere and one is the right hemisphere. This is corpus callosum. Okay, if you open the brain like this, you can see corpus callosum. This is falx cerebri. This is falx cerebri. You can see the surfaces. Superolateral surface. Here this is inferior surface and this is medial surface. And here you can see the folds. Okay. The upper one is called gyrus and the lower one is sulcus. Then coming to the lobes of the cerebral hemisphere. We have four lobes: frontal lobe, parietal lobe, temporal lobe and occipital lobe. And we have three poles: frontal pole, temporal pole, occipital pole. Here you can see central sulcus, which is separating frontal lobe with the parietal lobe. And this is parieto-occipital sulcus, which is separating parietal lobe with the occipital lobe. This is the pre-occipital notch. It is the lateral fissure, which is separating temporal lobe with the frontal lobe. Here you can see sulcus, central sulcus or fissure of Rolando. Central sulcus or fissure of Rolando. And posterior ramus of lateral fissure or Sylvian fissure. This is posterior ramus of lateral fissure or it is also called Sylvian fissure. This is parieto-occipital sulcus. Okay, so you have three sulci: central sulcus, Sylvian fissure and parieto-occipital sulcus. Then here we have to draw two imaginary lines. Okay. So a little anterior to the occipital pole, the inferolateral border shows a slight indentation which is called pre-occipital notch. Okay. So the first imaginary line connects the upper end of the parieto-occipital sulcus to the pre-occipital notch. Okay. So upper end of the parieto-occipital sulcus with the pre-occipital notch. So this is the first imaginary line you have to draw. Then the second imaginary line is a backward continuation of the posterior ramus of the lateral sulcus to meet the first line. So this is the lateral sulcus, Sylvian fissure. Here you have to extend a line which is going backwards and touching the first imaginary line. This is the second imaginary line. We have to draw two imaginary lines. Why we have to draw? To separate, to clearly separate the lobes. So this is the frontal lobe, which is separated from the parietal lobe with the central sulcus. And here between the frontal and temporal lobes, you have lateral sulcus, Sylvian fissure. And between the parietal and temporal lobe, you have lateral sulcus and the extension, posterior ramus of the lateral sulcus. That is second imaginary line. And between the parietal and temporal lobe, which is separated from the occipital lobe by first imaginary line. which is extending from the parieto-occipital sulcus superiorly, and inferiorly it is connecting to pre-occipital notch. Then coming to histological structure of the cerebral cortex. What are the cells present and how many laminae are present? We have got 6 laminae. Okay, 6 laminae from up to down. The first one is the molecular layer. So it is it is showing like this when you stain with the Golgi stain, and it is like this with the Nissl stain, and it is like this with the Vigat stain. So we have molecular layer, external granular layer, external pyramidal layer, internal granular layer, internal pyramidal layer, and multiform layer. So all this is gray matter. Then comes white matter. So in the gray matter, we have 6 layers in the cerebral cortex, 90%. Okay, except in the allocortex, except in the old one, 10%. Then you have some cells in each layer. Those are in the plexiform or molecular layer. It has transverse fibers and some scattered neurons. And in the external granular layer, many stellate neurons are present. And in the pyramidal neurons, mainly pyramidal cells will be present and some stellate and basket cells will be present. In the internal granular layer, we have stellate neurons and outer band of veil larger. And coming to the ganglionic layer, in this it has giant pyramidal cells and inner band of veil larger. And coming to the last layer, multiform or polymorphic layer. That is, neurons of various sizes and shapes will be present, and it will merge with the white matter. So the bands of veil larger are nothing but the myelinated fibers. Here comes Brodmann areas of the brain. A Brodmann area is a region of the cerebral cortex defined by its cytoarchitecture. This concept was introduced by German anatomist Corbinian Brodmann in the early 20th century. Brodmann mapped the human brain based on the varied cellular structure across the cortex, and he identified 52 distinct regions and numbered them as 1 to 52. These regions or Brodmann areas correspond with diverse functions. So in the frontal lobe, we have precentral cortex and prefrontal cortex. In the precentral cortex, you can see this is precentral cortex and this is prefrontal cortex. You have superior, medial and inferior frontal gyrus. They are separated by superior and inferior frontal sulcus. You have all these Brodmann areas in the prefrontal cortex. So coming to areas in the precentral cortex. It has primary motor area number 4. Lies in the precentral gyrus. In these 6 layers of the cerebral cortex are present, and here wedge cells are present and is agranular, and it has topographic representation of our body. It is responsible for initiation of the voluntary movements. Next, coming to premotor area. It is composed of 6, 8, 44 and 45 Brodmann areas. Area 6 is present anterior to primary motor cortex, and it is also having 6 cell layers, and it also composed of similar topographic representation of the body as primary motor cortex. It coordinates the voluntary action of the area 4 with the extrapyramidal system. Next, coming to area 8, it is also called frontal eye field. It is for control of eye movements. Next, coming to area 44 and 45, they are Broca's motor speech areas in the dominant hemisphere. Movements of the structures, so it is helpful for movements of the structures which are responsible for production of voice and articulation of the speech. Next, coming to supplementary motor area. It is present on the medial surface of the frontal lobe. Topographic representation is present. Upper body is represented in the dorsal aspect and lower body is represented in the ventral aspect. It works with the premotor area and it is for attitudinal, fixation and positional movements. So what are the afferents and efferents of the frontal lobe? Afferents are from the adjacent areas, opposite hemispheres and from the thalamus. The efferents will be corticospinal tract or pyramidal tract, in which 30% are primary, 30% from the primary motor area, 30% approximately from the pre and supplementary motor area and 40% from the somatic sensory area. The collaterals from pyramidal tract will go to adjacent areas of the cortex, basal ganglia, red nucleus, reticular formation, vestibular nuclei, pontine nuclei and inferior olivary nuclei. Here you can see is the central sulcus. This is the frontal lobe. Okay, this is primary motor area, that is area number 4. This is premotor area. This is Broca's speech area. Okay, so here and here our body will be represented like this. Okay, so this largest one is the most, that means it is required for fine movements. So more area will be given to fine movements. Next, coming to areas in the prefrontal cortex. You have areas 9 to 14, 23, 24, 29, 32, 44 to 47. So here you can see 9 and some 46. Okay. Some areas are shown. So why it is important? It is for planned action and for higher functions and is the seat of intelligence and it controls the intellectual activities. What are the afferents and efferents here? The afferents here are from the dorsomedial nucleus of the thalamus. So these afferents will go to 9 to 12 and 44 to 47 Brodmann areas. And from the anterior nuclei of the thalamus, it will go to 23, 24, 29 and 32 Brodmann areas. And from here, the efferents will go to thalamus, tegmental reticular formation, pontine nuclei, caudate nucleus and mammillary bodies. Next, coming to the second lobe, that is parietal lobe. We have primary sensory area, secondary sensory area. Primary sensory area or S1, it is formed by 3, 1 and 2. It is having granular, that is stellate and medium sized pyramidal cells. And here also we have topographical organization, okay, of the body. In secondary sensory area S2, okay, in post central gyrus below S1 also. So in S1 and S2 we have topographical organization. Coming to sensory association area or 5. It is for passive or active rotation of joints. And area number 7 is for discrimination between the stimuli. And area number 40 is the higher association area. It is for stereognosis. So you have S1, below that you have S2. Both have topographical representation and they are granular, that is having stellate cells. And here the sensor association area is having 5, 7 and 10. 40, 5, 7 and 40. 40 is specially called as higher association area. So it is the topographic representation. Okay. What are the afferent and efferents to it? The afferents for S1 are from the thalamus, that is VPL and VPM, and for S2 it is the from S1 and from the thalamus. The efferents will go to S1 again and to from the S1 to corticospinal, corticobulbar and corticonuclear tracts. So they will form these tracts and to caudate nucleus and putamen, and they will also go to thalamus, tectum, pons and cerebellum. Coming to fibers, association fibers, they will connect the other cortical areas. They are helpful for somatic sensations. Then coming to commissural fibers, they are from axons of pyramidal cells from the layer 3. They will go to opposite hemisphere. Okay. So S1 will go to opposite S1 and S2. S2 will go to S2. So through commissural fibers. Then what are the functions of parietal lobe? The S1 localizes, analyzes and discriminates different cutaneous and proprioceptive senses. S2 is for perception of the sensation. 5 and 7, they will differentiate the relative intensity of the different stimuli. And 40 is for stereognosis. Okay. The inferior part of the postcentral gyrus is helpful for taste and general sensations from tongue. The angular gyrus is for tactile localization and discrimination, and it will detect the extent and direction of movement of small joints. Then coming to the third one, the temporal lobe. It has primary auditory area, it is 41 and 42. They are present in the superior temporal gyrus. The afferents will come from MGB, that is medial geniculate body and pulvinar nucleus. Efferents will go to medial geniculate body, pulvinar and superior colliculus. It detects the loudness, pitch, source and direction of the sound. Then coming to auditory association area, that is area number 22 or Wernicke's area. Okay. And 21 and 20. So this 22 is present in the posterior part of the superior temporal gyrus. It will understand the meaning of the word and comprehension of the word. Area number 21 and 20, they are present in the middle and inferior temporal gyrus. They are for interpretation and integration of the auditory impulses. Then coming to the last lobe, occipital lobe. It has primary visual cortex or striate cortex. Area number 17 or V1. Okay. The afferents will come from optic radiation. The efferents will go to cerebral cortex, okay, superior colliculus and pulvinar nucleus. It is helpful for perception of the visual impulses. Then coming to visual association area or peristriate area. Area number 18. And visual association area or occipital eye field. Area number 19. It is for recognition. These both are for recognition and identification of the objects based on previous memory. And 19 is also for movement of the eyeball. Then the next video is about the white matter of the cerebrum.